Hello everyone, today we are discussing about malaria. Malaria is caused by a parasite which is called as Plasmodium. We will discuss the life cycle, clinical feature and how to manage a case of malaria. So the causative agent is Plasmodium and it completes its life cycle in two hosts. First is the men, which is intermediate host, asexual development of parasite occur in men, and second is the mosquito, female Anopheles mosquito, and sexual development occur in the mosquito. So, in the human cycle, we see that the sporozoite is the infective form of the malarial parasite, and these sporozoites are present in the salivary glands. So, with the bite of mosquito, sporozoites will enter the human body and they will travel all the way to the liver cells. And sporozoites multiply to form a schizone there in the liver cell itself, and 20,000 to 50,000 merozoites are formed. Upon development and upon maturation, the liver cell will get destroyed and there will be release of merozoites in the blood circulation. So the phase of pre-erythrocytic schizogony, which occurs in the liver, differ for different species, like 8 days for 5x, 6 days for falciparum, 9 days for ovale, and 13 to 16 days for malariae. After entering the circulation, these merozoites will attack the RBC. Inside RBC, they will pass through the stages of trophozoite, then schizone, then merozoites. So trophozoite is very specific because it look different for different species. We will discuss that. And these merozoites also contain male gametocyte and female gametocytes. So merozoite can attack new RBC or they will be picked up by mosquito and further development will take place. So let's first discuss the trophozoite and schizone of different species. Plasmodium vivax ring form, falciparum multiple ring forms, malariae ring form which look like this, and ovale ring form with dots in the cytoplasm, and schizones of different types. Then coming to the life cycle, we have seen like mosquito will bite. First bite of mosquito will lead to a sporozoite entering into the human being, then pre-erythrocytic or exoerythrocytic cycle in the liver, merozoite will be released, and erythrocytic cycle in the RBCs, and it will develop into trophozoite, schizone, merozoites, and these merozoites are taken up by the blood meal by mosquito, and then further development, which is sexual development, like macro and micro gametocyte will form macro and micro gametes, and then on fertilization they will form zygote, ookinete, and then oocyst. Usually asexual and sexual both are taken up by the mosquito, but only mature sexual forms will further develop. So oocyst will be formed, and then oocyst will increase in size and form hundreds and thousands of sporozoites. Sporozoites will be distributed in the body cavity of the mosquito, and as it has more predilection for salivary glands, it will be deposited there. And with the bite of mosquito, these sporozoites will enter again into the men and cycle will go on. So here is a picture of different gametocytes of vivax, falciparum, malariae, and ovale. Now coming to the clinical features of malaria. First is febrile paroxysm. It occurs in 3 stages: cold stage, hot stage, and then sweating. In cold stage, patient experience cold and shivering. In hot stage, patient experience high fever and severe headache, nausea, vomiting, followed by sweating. So there is a hint that fever misses a day. And second is the anemia, because there is destruction of RBCs, so there will be hemolytic anemia after a few febrile paroxysm. Then destruction of RBC will lead to splenomegaly as well, so spleen will be enlarged and palpable, and also there will be thrombocytopenia, and DIC can occur in severe cases. Coming to the diagnosis, we will order a PBS, peripheral blood smear, both thick and thin film stained with Giemsa or Leishman stain, and examine under oil immersion. So thick blood smear will detect the parasite, it is the best test, and thin blood smear will detect the species. If thick smear is negative, we will repeat the smear 3 times every 12 to 24 hours, and if it is negative, then we rule out malaria and look for other causes of fever. Coming to the lab test, CBC, Hemolytic anemia will be seen, as we have discussed that there will be destruction of RBCs. So hemoglobin will be decreased, platelet will be decreased, LDH will be increased, and possible leukocytopenia. Third is fluorescent microscopy. Kawamoto technique is used, in which blood smear is stained with acridine orange, and nuclear DNA will appear green and cytoplasmic RNA will appear red. Then comes the QBC, quantitative Buffy coat. In this, blood is taken as in a QBC tube with fluorescent dye and centrifuged, and then the Buffy coat is examined under the fluorescent microscope for parasite. It is very expensive test, so it is not done. Coming next to the rapid diagnostic test, which is very common, it detects the antigen using immunochromatographic methods. So kits are available to detect the HRP2, which is histidine rich protein 2 of Plasmodium falciparum. This is also called as parasite F test. PLDH of all the 4 species can be detected. Other tests are serology, DNA probes, PCR. So that is it for today. We will discuss the treatment aspect in another video. And hope you like the video and subscribe to my channel, The Medical Doctor.